Okay, I'm going to talk about inherited change, meiosis specifically. We perform meiosis because we need to reproduce. So meiosis leads to our gametes. We form 4 haploid cells, and a haploid cell is a cell with one complete set of chromosomes. So it's got 23 chromosomes instead of 46. And okay, the stages are, so in meiosis you have meiosis 1 and 2, and they're quite similar to mitosis, but slightly different. So if we start over here with prophase, you have like the early, middle, and late stages, and what happens here in the first stage, the chromosomes condense, they replicate, and then you go on to form those homologous pairs. They pair up, and the centrioles, they duplicate. So those are the things that will form spindles later. The nuclear envelope disintegrates, and we go into metaphase 1. Metaphase 1 is where the spindles form, and the homologous, it's the homologous chromosomes that pair up together, and each chromosome is connected to one of the spindles on one side, and the other one's connected to the other side, so that later when you see an anaphase, the whole chromosomes, the pairs get actually split up, and not the 2 chromatids, like in mitosis. Here also what happens is crossing over. So crossing over is what we do so that we can enhance variation, and we just have something like this, and they sort of mix together. So at different points, this guy will swap a bit of his chromosome with the non-sister chromatid for this bit. So like you can see here, some of the stripes have gone here and some of the dots are here. Then going back to this, you have anaphase, they divide, and then telophase, which is where the nuclear envelope forms again. And in animals, cytokinesis does actually occur, so the 2 cells will separate out. In plants, it doesn't. Then we go to prophase 2. So what's interesting about this now is there is no interphase between telophase and prophase 2, so the chromosomes aren't replicated again. And basically you just go through 4 stages, which are really similar to mitosis. You have in prophase 2, the nuclear envelope disintegrates. You go to metaphase, and they are pulled apart, and then you go to they line up together, and then in anaphase 2 they're pulled apart, and then in telekinesis, in telophase, the nuclear envelopes form, and cytokinesis occurs, and you end up with 4 haploid cells. So because we didn't replicate the DNA here, you have half the number of chromosomes than you usually would after, for example, mitosis. Cool.